Okay. Hey, thank you to the organisers for letting me speak today. So I'm a PhD student from Helen Sable's lab at Birkbeck, and I'm just going to be talking about the structure and function of the Merozoic surface protein 1 in the malaria parasite Plasmodium falciparum. So malaria is still a global health problem, causing around 400,000 deaths per year. And there are various Plasmodium species that can cause malaria, but the species responsible for 90% of mortality is Plasmodium falciparum. This is an apicomplex parasite, which has a multi-stage life cycle, involving a stage in the mosquito midgut and salivary glands, as well as the human host, liver, and red blood cell. And it's this red blood cell cycle which is responsible for most of the parasite's pathology, and where I'll be focusing mainly. So just to go through that in a little bit more detail, the invasive form of malaria, the merozoite, invades the red blood cell, undergoes various stages of DNA replication and organelle synthesis, and then then to form this multinucleated cell known as a schizont. The schizont then the organelles and nucleus then segment into individual daughter merozoites, and then this is the point at which the process known as egress is initiated, which is where the daughter merozoites burst from the red blood cell. And so essentially what happens is the parasitophorus vacuole in which the malaria develops within the red blood cell rounds, the merozoites form a flower sort of formation around what's known as the food vacuole, which is where the byproducts of hemoglobin digestion are deposited. And then the parasitophorus vacuole membrane porates and degrades. The red blood cell membrane collapses and porates, and then there's finally rupture of the red blood cell membrane. And I just want to draw your attention to this drug here, E64, which is a cysteine protease inhibitor, essentially prevents red blood cell membrane rupture. So now just to quickly talk in more detail about the merozoite, the invasive form of malaria in the red blood cell cycle, and specifically its surface coat. So on the right here, you can see electron micrograph of two merozoites. Here are the two plasma membranes of the merozoite, and you can see it's heavily decorated with proteins. And this is the surface coat, and the major component of this surface coat is merozoite surface protein 1. So MSP1 is a complex. It is a GPI-anchored protein, which is what ensures it's tethered to the merozoite surface membrane, and it's still of unknown function. It's normally associated in complex on the surface with another merozoite surface protein known as MSP7. And upon initiation of egress, it's processed by a malarial serine protease known as sub1. And it's processed at three different sites, the H330, 38, and 3842 site. And these are just named because of the size of the fragments which are produced upon processing. And this 3842 site is particularly important, has been shown to be particularly important for parasite viability. And there are three alternative cleavage points that the enzyme can recognize at this site. So once processed by sub1, all the fragments of MSP1 remain non-covalently associated on the merozoite surface. And this is then thought to allow other merozoite surface proteins to bind, which may be important for MSP1's function. So now I'm just going to go into a bit of cell biology. Hopefully it's not too boring. So MSP1 has thought to have a role in invasion initially, but now we believe it might be involved in egress, which essentially is this process of merozoites bursting out of the red blood cell, as I've mentioned. So within our lab, we managed to produce a parasite line which lacks MSP1. So you can see on the left here is the situation in wild type. So blue highlights the plasma membrane, and then the yellow arrows highlight MSP1 on the surface. And then in the MSP1 null, we have this smooth plasma membrane. And essentially what we saw in this MSP1 null line is an egress defect. So if you look at the videos on the left is the wild type situation where the merozoites burst sort of explosively out of the red blood cell, and then the MSP1 null has defective egress. Merozoites don't disperse normally, and egress seems sort of stunted. So when MSP1 is completely absent from the meroite surface, there's an egress defect. And there are various possible explanations for this. So maybe MSP1 is involved in degradation of the parasitophorus vacuole membrane, or maybe it's involved in red blood cell membrane rupture, or maybe it simply acts as a scaffold to recruit other proteins to the merozoite surface for egress. And so we then looked at the MSP1 null line parasites to see why this egress defect might be occurring. And essentially what we did was we looked at it prior to egress, so treated with that cysteine protease that I mentioned earlier that prevents red blood cell rupture, or after egress, so after the red blood cell membrane had ruptured. And in the wild type, just prior egress, the parasites are properly segmented from the food vacuole, which is here, and inside the hemozoin, the byproduct of the hemoglobin digestion. And the parasitophorus vacuole membrane has degraded as expected. And then just after egress, you can see red blood cell membrane is ruptured, and merozoites are sort of dispersed from the residual body, which is the remnants of the food vacuole. But in the MSP1 null line, we saw this segmentation defect in which merozoites' contents appeared to not have been segmented or organized properly, and still sort of remained attached to an extended food vacuole. But the parasitophorus vacuole membrane had burst. So there were remnants seen of that membrane, suggesting that this still had occurred. And then after egress, the red blood cell membrane rupture had occurred, but you still were seeing this extended residual body in the remnants of egress. So we did some quantification of this, and essentially in the 40% of cases where the food vacuole was visible in pre-egress cells, so that's the E64 treated schizonts, 89% of the MSP1 null parasites had not segmented properly from the food vacuole in comparison to 14% of the wild type. And in both cases, there seemed to be similar levels of rupture of the parasitophorus vacuole membrane. Whereas in the post-egress cells, in the 40% of cases where the food vacuole was visible, again, we saw that the merozoites in 60% of the MSP1 null parasites had not segmented properly in comparison to 17% of the wild type, but in both cases, similar levels of red blood cell membrane rupture. So from this, we kind of thought that essentially, because when MSP1 is absent from the surface, this vacuole membrane and red blood cell membrane rupture are still occurring. Maybe MSP1 has a role in this, causing this segmentation of merozoites from the food vacuole. But this might be a sort of artifact of loss of the GPI anchor, which tethers MSP1 to the plasma membrane, changing the curvature of the plasma membrane or its fluidity, therefore preventing proper segmentation. So maybe simple removal of MSP1 doesn't tell us much about the function. And so just to conclude this kind of cell biology, essentially we wanted to look at ways of probing the function of MSP1 without completely eliminating the protein. And so because we knew this 3842 cleavage site of the serine protease sub1 had been shown to be important in parasite viability, what we did was we created a mutant that has this site completely eliminated based on what's known of the enzyme's preferences. And here on the, just at the bottom, we put wild type and mutant cells together and watch them egress. And so here wild type's labeled with DAPI and mutant is unlabeled. And you can see again, this egress defect was still seen. So wild type bursting normally, and then mutant with stunted egress. And essentially in 80% of wild type cells, egress was occurring normally, but only in about 40% of mutant cells was egress occurring normally. So maybe inhibition of correct MSP1 processing by sub1 results in an egress defect, but we wanted to look at the molecular structure of MSP1 both before and after sub-1 processing to see if this indicated anything about MSP1 function in merozoite egress. So in order to do this, what we did was expressed a parasite line in which, in place of the GPI anchor on MSP1, we had a flag tag. And this allowed us to purify MSP1 in both unprocessed and sub-1 processed forms, because essentially in mature schizonts, we just would lyse the cells to get unprocessed sub-1 before unprocessed MSP1 before egress, or sub-1 processed MSP1 was collected after cells had egress from the culture supernatant. So first looking at unprocessed MSP1. So what we did was, Natasha helped make some cryogrids and took about 10,000 movies on the Krios. And the particles were barely visible, even in ice, but we managed to pick them by blob recognition in Cryospark, and then got some nice 2D classes. And finally, after further cleaning of data, a 4.1 Armstrong model, a map, sorry. And then we looked at the sub-1 processed MSP1, so this is after egress. And we looked at 20,000 micrographs. Again, the particle is barely visible. Picked particles with Cryolo, then got some nice 2D classes with a few more orientations than the processed, but more heterogeneous data. We did some 3D reconstructions in Reliant and saw that data was indeed heterogeneous. So like there were different reconstructions with additional density. And because we're looking for partner proteins on MSP1 to sort of suggest things about its function, we took forward this class with the additional domain and managed to refine it to about 4.4 Armstrong. So whilst we were doing this about a year and a half ago, the structure of a recombinantly expressed sub-1 processed MSP1 was published. This was solved by cryo-EM to 3.2 Armstrong, and essentially showed that MSP1 was made up of 37 alpha helices, and it has this wing domain and also a stalk domain, and the stalk domain is made up of the 3842 kilodalton fragments, and the wing domain is made up of this H3 and 30 kilodalton fragments. So we used this structure and mapped it and docked it into our map to see if our MSP1 that had been purified directly from parasites had additional partners present. And it did. So in the uncleaved MSP1 and the sub-1 cleaved MSP1, we saw additional density on the wing domain, as well as in the cleaved MSP1, additional density on the coil domain and the back of the coil domain. Where we were able to, we did see alpha trace of this additional density, and saw, although we were unable to trace this this density here because it was very flexible, we were able to trace on both structures this density on the wing. And looking at other proteins, looking at AlphaFold predictions of merozoite surface proteins, we managed to find a match to this C alpha trace of the additional density in the map. So MSP7, the C terminal helical bundle, perfectly overlays with our C alpha trace of the additional density in the map, suggesting that MSP7 is both present in the uncleaved and cleaved MSP1. And so now we need to try and identify what this density at the bottom of the coil domain to help understand what sub-1 processing is doing to MSP1 and what the function of MSP1 is in merozoite egress. So thanks. Just to summarize, MSP1 is a major component of the merozoite surface coat. Knockout of MSP1 impairs egress, but rupture of the parasitophorus vacuole membrane and red blood cell membrane still occur. And so maybe knockout and so maybe MSP1 is involved in merozoite, knockout of MSP1 causes segmentation defect. But this may be due to the loss of the GPI anchor. So the function of MSP1 is still unclear. And we've also proven that loss of the 3842 cleavage site of sub1 on MSP1 also impairs egress. And the published structure of recombinant MSP1 corresponds with our structures of native MSP1 complex, and partner proteins are present in our map. So yeah, thanks to everyone in Helen's group, past and present, and the group at the Francis Crick, who helped me with a lot of malaria culturing, as well as people at CUI and the EM lab in Birkbeck and computing. Thank you.